All right, so really excited to be here. So I would like to share our current progress on what the title says, assessing microbial genome representation across various databases. So I'll try to be quick so we guys will try to be on time to the coffee break. So obviously, as you heard from the speaker today, I don't need to define the metagenomics analysis, but in case I do, so primarily for taxonomic profile, we would like to know who is there and which abundance is present. And this is the typical pipeline, which you also probably know. And then one particular thing we are curious to know, and I didn't hear much today, is about the reference genome databases, right? So besides kind of paying attention to the method, which is taking the metagenomics trees and try to predict who is there, it's also very, very, very essential to know, to have the reference. And this is where we're trying to make contributions. So my claim is if we don't have the accurate reference, perhaps the analysis we'll perform will be not accurate. If we have incomplete reference, that's also the problem because we will be never discovering what is not in the reference. So what I'm trying to say, it's very, very important to pay attention. And generally, the microbial reference databases should be high quality reference genomes with annotations for the known species. And will help us to perform the good taxonomic profile. And however, the reality is we don't necessarily all the time have the full genome in the reference. Very often time is fragmented, and there is some discrepancies across databases. So if you take the current databases you have and just looking at the number of species they have, you will probably notice something's happening there, right? You can see the range between 20,000 to almost 100,000. And then just look at those numbers, you will perhaps think that some of them are more complete or more noisy, in case you don't believe that what is there is correct. But definitely there are differences. So if you just consider the names of the species present across the databases and you will do sort of Venn diagram, you will see that many species are present only in one database, sometimes two databases, but quite rarely present across three databases. So in this case, we look at the bacterial species and we look across three major databases like Patrick, RefSeq, and Ensemble. So this is and this is the link to the paper a while ago. So, and we have recently updated the results. So if you ask the question, maybe that just happens to bacteria, but doesn't happen to viruses and fungi, that will be not the case. The same story happens to the fungi, for example, where there is a few species present across all the databases. So in that case, we never touched the genomes yet. All we have looked at, we just looked at the taxa ID and the names of those species. And this is how it started, and this is the paper we published before. But let's have a look at something in common. If we have a species present in two databases, will the genomes of those species be the same? So oftentimes will be fragmented. So perhaps there is a true genome, which is full, and then each database will be presented as the contigs, and contigs will disagree or agree to some extent to each other. And the interesting question becomes, should we or should we not try to merge the contigs? Because what we don't know the true genome. However, we know the contigs from database 1 and we know the contigs from database 2. So what is the approach to take? So we are on the way to develop the approach. We don't have an approach yet, but we observe many interesting cases, which are quite intriguing and makes us think what should we do. So the first step we would like to do, we would like to say how many times that happens, how many times the genomes across the database for the same species are different. So this is one example. We have looked, I think I didn't put the name of the species, but as you can see, there is some parts unique to each database. So this is RefSeq on the top versus the Patrick. So there is a unique piece in RefSeq as well as there is a unique thing in Patrick. So now we're thinking, what is the best way to assess the difference? So far we thought about, let's take maybe total match and divide by the length of the longest genome, or maybe the union of genome. That's something we think. So if you take the longest genome, that will be around 30%. So what 1 minus, so that will be indicated that the two contigs across the genome are different by 30%. It's another example from Shannemela, I think Schwadebella, pronounce that correct. So you can see that there are some parts identical, some parts are not identical. There are some parts being unique to one database. And then the question becomes like, should we, if we think about kind of merging that or maybe creating some sort of master database, should we force to unite it like that? Maybe it's a good idea, but however, we might be forcing that too much to the degree that we don't know if you go back for a second, we don't know what the difference is. It the unique kind of structural variance from a particular strain from one database? Is it the sequencing error? Is this a bioinformatics error? We don't know. So the question becomes, should we or should we not merge? So the problem formulation, we are suggesting given a set of contigs for the species S in database A and the species in database B, we want to do two things. We want to compare the genome representation between A and B and device similarities and then the differences and kind of quantify that. And I think ultimately we would like to produce merge contigs in a sort of master database across all the databases. So this is the workflow we do. We download the data, we compare the databases, we process genomic sequences, and we process file names. And then eventually we compare genomic sequencing and report similarities and differences. So if we just focus, I would like to share some preliminary results. So if we just focus on the full genomes, they I think look a little bit better. So we took full genomes, and the condition of the full genome is that there is a single contig as well as should be labeled as a complete genome. So we look at the complete genome, and there is a little bit above 13,000 complete genomes, which are common between the Patrick and REF6. So we have looked at that. We have excluded the genomes which are not available. Sometimes there is some technical issues and some broken links and other kind of things which we are not able to access genomes. So we take the one available. So there is quite small percentage of genomes, 1%. So it's 116 genomes which are not identical across the databases. Look at that. On 116 genomes, which are not identical, we see that some of them are quite similar, with this tiny fraction being different. However, others are more distant from each other. So those are kind of all we have at the moment. So we actively work on that on two kind of components. So one, defining the right metric to quantify the differences, and another one to think about what is the way to merge those genomes. So for the future direction, we would like to quantify redundancy uniqueness between the widely used bacterial reference genome, measure similarity, and eventually would like to create a master database, which we think will be better to use than each individual database. So that's work of many people in the lab, primarily Grigory Boldarev, which is not on the picture, but is and we're looking for the postdocs if you're interested to work on the similar project as here. Talk to me, and yeah, we'll be, hopefully I did it quick, so we are okay for the coffee break. Happy to take questions. Thank you.
Thank you.